Welcome to this explainer. Okay, today we are going to dive into the absolutely fascinating hardware that powers some of the most critical, life-saving tests in modern medicine. We're embarking on a molecular journey, really unpacking a technology that literally bridges the gap between complex biological fluids and incredibly precise, life-altering data. We are talking, of course, about liquid chromatography-mass spectroscopy, or LC-MS.
Right off the bat, let's look at the incredible clinical stakes here. The goal? Identifying one specific molecule hidden inside a messy, complex biological soup to detect early indicators of disease. I mean, think about this for a second. Using just a single dried blood spot, a modern LC-MS system can screen a newborn for more than 20 inborn errors of metabolism. We're talking things like phenylketonuria or sickle cell anemia, and it does this in under 2 minutes. 2 minutes! That rapid, multiplexed analysis allows for immediate, life-saving interventions. It really is a total marvel of analytical chemistry.
So, here's our roadmap for today. We'll cover 1. Life-saving clinical diagnostics. 2. Separating complex biological mixtures. 3. Solving the vacuum challenge. 4. Quadrupole versus TOF analyzers. 5. Minimizing interference and suppression. And finally, 6. Proteomics and automated futures. Let's jump right into Section 1, Life-Saving Clinical Diagnostics. To truly understand the magic here, we have to follow the physical journey of a sample, step by step, from the moment of injection all the way to the final data output. First up, the sample gets injected into a high-pressure stream. From there, it's pushed right through a liquid chromatography column to separate the mixture out. Next, it hits the ionization interface. This is crucial because it converts the sample into gas phase ions. Those ions are then sorted in the mass analyzer and finally interpreted by the data system.
Now, if you look at the scientific blueprint of this machine, you can clearly see how the pressurized liquid side, the LC pump and the inlet, physically connects to the high vacuum mass spectrometer. And honestly, for decades, combining these two techniques was a massive headache. It was hindered by a fundamental physical incompatibility. You simply can't pump high-pressure liquid straight into a high-vacuum chamber without completely destroying the system. The bridge that finally solved this is the atmospheric pressure ionization, or API source, which we're going to explore in just a bit.
Moving on to Section 2, Separating Complex Biological Mixtures. Before we can weigh these molecules, we have to isolate them. The HPLC hardware acts as a brilliant separation engine for these complex biological mixtures. It pumps the sample through an analytical column with incredible sensitivity. And guys, this isn't just gravity doing the work. Modern systems use solvent delivery pumps that push liquid at extreme pressures, sometimes reaching up to 1200 bar. The sample is forced through a column packed with microparticles coated in specific chemicals. Depending on a molecule's chemical properties, like its polarity or hydrophobicity, it interacts differently with this stationary phase. Hydrophilic compounds might just flush straight through, while hydrophobic ones tend to stick and take longer. This differential migration is absolutely essential because it ensures we send a perfectly sequenced stream of individual analytes into the detector, rather than just dumping a chaotic bulk mixture all at once.
All right, Section 3, Solving the Vacuum Challenge.
So, as a scientist, you know the mass spectrometer must operate under a high vacuum, right? Otherwise, your ions just collide with ambient air molecules and scatter. Atmospheric pressure ionization, or API, elegantly solved this dilemma. By simply moving the ionization process outside the vacuum, the system can comfortably handle those high liquid and gas volumes coming right off the HPLC. The liquid gets nebulized, the solvent evaporates away, and the analyte is given an electrical charge just before it's carefully drawn into that high vacuum chamber.
Let's look closely at the real hardware mechanics of two key API sources. On the left, we've got atmospheric pressure chemical ionization, or APCI. Here, the eluate is sprayed through a vaporizer that is heated up to a blistering 500 degrees Celsius. Then, a corona discharge needle emits electrons to ionize the solvent, which in turn transfers that charge to the analyte. Now on the right, we have atmospheric pressure photoionization, or APPI. Instead of a needle, it uses a vacuum ultraviolet lamp to generate photons. This is a fantastic workaround for ionizing highly nonpolar compounds that stubbornly refuse to take a charge through the usual methods.
This table is a great cheat sheet for how different ionization interfaces adapt to specific chemical challenges. Electrospray ionization, ESI, is the absolute gold standard for proteomics. It uses a strong electric field to create charged droplets, which makes it ideal for polar, high molecular weight labile proteins. It even creates a multiple charging phenomenon so massive proteins can carry dozens of charges, scaling them down into a measurable range. But ESI really struggles with nonpolar compounds. That's exactly where our other tools step in. APCI is perfect for moderately polar volatile and thermally stable molecules like lipids. Meanwhile, APPI is your highly specialized tool for nonpolar, neutral analytes.
This brings us to Section 4, quadruple V's TOF analyzers.
Once ionized, the analytes enter the mass analyzer to be sorted. Let's contrast the targeted precision of quadrupole mass filters with the sheer discovery power of time-of-flight analyzers. A quadrupole is essentially 4 parallel metal rods that use direct current and radio frequencies to filter ions. A triple quadrupole system is incredible for targeted analysis workflows. Think routine clinical drug monitoring. It locks onto a specific precursor ion, fragments it, and measures a specific product ion in an MRM workflow, which drastically cuts down background noise. Conversely, time-of-flight or TOF analyzers measure the exact time it takes for an ion to travel through a flight tube. Lighter ions just fly faster. TOF gives us high-resolution accurate mass and captures all ions in a single run. This untargeted approach is an absolute game-changer for metabolomics when you're casting a wide net to discover completely unknown compounds.
Section 5. Minimizing interference and suppression.
Here's a common hurdle. If you have too many background matrix components from your biological sample, like competing salts or lipids, they can actually suppress the ionization of your target analyte. To fix this, here are some quick practical tips. First, implement enhanced chromatography separation, so your analyte elutes far away from the interference. Second, utilize solid phase extraction during sample prep to strip out unwanted proteins before injection. Sometimes, honestly, a simple dilute and shoot method does the trick just by lowering the overall concentration. And crucially, always switch to volatile buffers like ammonium acetate. If you use non-volatile buffers, you are going to contaminate your pristine ion source almost instantly.
And finally, Section 6, proteomics and automated futures. So how does this hardware build the future of precision medicine? Well, by incredibly isolating low-abundance proteins, LC-MS aids in identifying very early indicators of disease through proteomics. Researchers literally digest proteins down into peptides and sequence them using fragmentation to uncover hidden disease signatures. Plus, in clinical trials, LC-MS is the absolute backbone for quantitative bioanalysis. It's used to rigorously monitor drug absorption, evaluate pharmacokinetics, and track drug metabolism, making sure new therapies are safely processed by the human body.
But the sheer volume of data these ultra-high-resolution systems generate is staggering. We are now witnessing a rapid transition toward much faster, automated solutions. Bioinformatics tools and machine learning are stepping in to automate the complex data annotation for researchers. This is especially vital in biopharmaceutical testing. By integrating machine learning-processed LC-MS data with electronic health records, scientists can correlate specific molecular signatures directly with patient outcomes. It's totally revolutionizing the workflow.
As LC-MS continues its incredible molecular journey, from a raw drop of blood, pushed through high-pressure columns, blasted by a 500-degree corona needle, all the way to machine learning-driven analysis, we really have to wonder, with all this automated power, what groundbreaking hidden biological secrets will this machinery unlock tomorrow? The precision of this hardware is quite literally rewriting the boundaries of modern medicine. Thanks for joining me on this explainer, and keep questioning what's possible.